Good afternoon or good morning, depending upon which time zone you're located in, but welcome to another one of our webinars on improving the relevance of drug interaction warnings. Today we're going to talk about a drug interaction involving potassium-sparing diuretics and ACE inhibitors and ARBs, angiotensin receptor blockers. We hope this seminar is useful for you in your practice, and hopefully it will help potentially reduce alert fatigue at the same time and improving patient safety. So I want to thank the team that has helped put together not only this seminar, but other seminars. We have individuals from the University of Arizona, where I'm located. I'm Dan Malone, I'm your host. University of Arizona, University of Pittsburgh, and the University of Washington. So thank you all for your help on this project and continuing efforts. So I'd like to just provide just a quick overview of the webinar, and then we'll turn it over to our speakers for today. So first, in terms of conflict of interest, they're demonstrated here on this slide that generally speaking, most of our presenters have no conflict of interest. Dr. Horn and Dr. Hansen, publishers of Hansen and Horn Drug Interactions Analysis and Management, the top 100 drug interactions, a guide to patient management. This grant was supported by, or this work is supported by a grant from the Agency for Healthcare Research and Quality, although the content is the sole responsibility of the authors and does not necessarily reflect the views of the Agency for Healthcare Research and Quality. So now we kind of have some of the do's and don'ts covered. This is a slide that we use to represent what we're doing. So we know that we have many slices of the defenses to prevent drug interactions from reaching patients. And that this includes not only the prescriber's knowledge, but also how we build our computer systems, the pharmacist, the patient's susceptibility to the particular drug interaction, how we administer the drugs, patient education monitoring. It's only when we have a rare occurrence where we have a failure, which are the holes in this particular model, that we end up getting a adverse drug reaction occurring, because the patient was exposed to both. So today we want to talk a little bit about determining to what extent that we have an adverse drug reaction. And we're going to be talking about this particular flow diagram where we look at the combination of an ACE inhibitor or an ARB in combination with the potassium-sparing diuretic. And the majority of our time we'll spend talking about how we reach conclusions about at what point in time do we want to provide a warning to a prescriber about the potential problems that a patient might have. And here the issue is hyperkalemia. So one of the things that we'd like to do is give you some data that supports kind of our cut point for this threshold of 5 millimoles or milliequivalents per liter within 48 hours. It gives you a sense of the evidence that goes behind that and why we think that this algorithm is probably a good way of approaching this particular drug interaction warning. What I'd like to do next is turn it over to Dr. John Horn, who's a professor emeritus at the University of Washington. And John is going to present to us the evidence associated with hyperkalemia and exposure to these medications. So John, how about I turn it over to you?
Thanks, Dan. You can move on to the next slide. I need to look at my picture. We'll look at this picture instead. I just want to take a minute or two to just kind of remind everybody what the potential mechanisms are involved with this interaction between the ACE inhibitors / ARBs and potassium-sparing diuretics that we're going to be talking about today. This graphic out of Palmer's article is a nice one. There's others that are very similar to it. But as you can see, there's a number of different places where drugs can get involved in the potassium homeostasis. And that most often occurs by alteration of the renal handling of potassium, or specifically the excretion of potassium by the kidneys. Of course, if you give a drug that's got potassium in it, like KCl, you can increase potassium supply. And there are a few disease states, for example, that can shift potassium in and out of cells and also change potassium homeostasis. But we're going to really focus today on the mechanisms associated with the drugs in question. And there are a number of different mechanisms here, as you can see. The ACEs and ARBs can produce hyperkalemia actually through multiple mechanisms when you consider both classes of drugs. All of these drugs affect the renin, angiotensin, aldosterone system, and the point in that system where the drug works is illustrated on this particular graphic. You can see the ACE inhibitors, of course, block the conversion of angiotensin 1 to angiotensin 2. The ARBs interfere with the receptor on the adrenal gland. And farther on down the pathway, we've got aldosterone blockers where we find the potassium-sparing diuretics. And then there also are effects of diuretics, particularly some of the potassium-sparing diuretics, again, that block sodium channels in the collecting duct. So the mechanisms here are very interesting because this truly is a pharmacodynamic interaction. There's no changes in the concentrations of any of these drugs due to the interaction, but there's changes in the way the body responds, the pharmacodynamic response to the drugs. And the thing that makes this particular interaction a bit unique is that there are multiple mechanisms involved, so that when we talk about a ACE inhibitor and a potassium-sparing diuretic, both of which can produce elevations in potassium, they do it by different mechanisms. Oftentimes, pharmacodynamic interactions are occurring when drugs have a very similar mechanism, in fact, acting at the same receptor. We don't have that in this case, and so therefore we don't have the limiting effect of two agonists working at the same receptor. And when that happens, what you inevitably get is essentially one of the two agonists acting as a competitive antagonist to the more potent agonist. And that tends to blunt the response. But we don't get that with these pairs of drugs. And so we don't have that sort of built-in pharmacologic blunting, if you will, that occurs with other pharmacodynamic interactions. Let's go to the next slide, please, Dan. This is a summary slide just looking at some of the various features, diseases, and drugs that can affect potassium. And here we're really focusing on hyperkalemia. And I'll let you quickly read the slide. I don't need to read it to you. But the point here is that there is a lot of various both disease states and drugs that can influence potassium homeostasis, as we've mentioned. And as I said, we're going to really focus on the drugs associated with this, and in particular, the ACE-ARB and potassium-sparing diuretic combination. Let's go on to the next slide, please. Now, if we think about angiotensin-converted enzyme inhibitors and ARBs, there are actually a number of different pharmacodynamic interactions that occur with these. The one we're going to focus on, of course, is the first bullet here, the increase of potassium concentrations. But there have been other interactions studied as well that we're not going to talk about today, but just for your inclusion, we've added some of the more important ones here. You can see increased lithium concentrations with these drugs. NSAIDs markedly reduce the hypotensive effectiveness of these drugs. And then because some of these drugs are prodrugs and require metabolism to an active metabolite, which is predominantly done by 2C9, 2C9 inhibitors can actually reduce the effectiveness of the ACEs. And so let's move on to the next slide. Okay, so the three classes of drugs that we're going to talk about are listed here for you. And if you look at the effects of any single drug in any of these classes, you find that all of these have the potential to elevate the potassium. For the potassium-sparing diuretics, that's typically in the neighborhood of a 0.2 to 0.3 millimole per liter increase. For the ACEs and the ARBs, given again alone, you see somewhere in the 0.1 to maybe 0.4 millimole per liter increase. So taken by themselves, these compounds can increase serum potassium, but usually not to the point where patients can develop hyperkalemia and therefore have some adverse outcomes. Let's go to the next slide. And take a look at hyperkalemia. This is usually identified as having a serum potassium level which is greater than 5 milliequivalents per liter or 5 millimoles per liter, same values. Those are the only equivalents I know, is the potassium milliequivalent and millimoles are the same. But anyway, drugs associated with this, as we've mentioned, include a number of other compounds, and we've included some of the more common ones here. The mechanisms are all a little bit different. We've got some direct-acting renin inhibitors like NSAIDs and the calcineurin drugs and aliskiren. Trimethoprim is an interesting drug. It's an antibiotic, as you know, and it looks to the kidney just like amiloride, which is, of course, a potassium-sparing diuretic. And we'll talk a little bit later about some data with trimethoprim. And then finally, there are drugs that increase potassium intake, and those are the potassium supplements that are used for that. But in sort of the outpatient patient delivery side, such things as salt substitutes and, of course, foods that are high in potassium can elevate serum potassium. Diseases associated with hyperkalemia, the primary one that we think about is reduced renal function because, of course, the kidney is the primary organ of excretion of potassium. But other other diseases such as diabetes, heart failure, and advancing age, which is not a disease, I know, but all of those are also associated with hyperkalemia. Okay, let's go to the next slide and look at a couple of case series that have been published. These are not really large ones, but they are important because they had a fair number of patients who had significant elevations of potassium, and in fact, in some of these cases, patients actually died from it. So bullet number 2 is probably the one I really want you to focus on. This particular interaction where we've got ACE inhibitors and potassium-sparing diuretics has been quite well studied. And what is found over and over and over again are these set of risk factors. And they are, as listed here, renal failure, diabetes, heart failure, age above 70, or taking other drugs that can produce hyperkalemia. And we've talked about some of those a minute ago. It's also been identified that the dose of spironolactone appears to be important in predicting the magnitude of the effect on potassium. There doesn't seem to be much data nor much evidence that the different drugs within the classes, in other words, if you look at all of the ACE inhibitors or all of the ARBs, is there differences amongst those drugs in their propensity to produce hyperkalemia? And if there is, we just don't have good evidence of that at this point. Let's move to the next slide, please. This is another small case series. And again, patients who had really pretty bad outcomes from this. And once again, we see exactly the same risk factors here. A lot of diabetes, a lot of renal dysfunction, and high doses of spironolactone. And again, patients in this case above 65 years of age being very common. So these two slides together, I think, are just really stating what the data shows when you look at a lot of the cases that have been reported with these agents. You see this very typically in these patients who have these pre-existing features, and not a lot in everybody else. So it's a useful thing to keep in mind. Now, if you go to the next slide, Dan, we were trying to find some evidence of how these things add together, and we couldn't find any. So thanks to Dr. Hanston, we made it up. So this is a hypothetical slide. So there's no data to support this other than what we think is going on. And what we tried to show here is the incidences of symptomatic hyperkalemia, asymptomatic hyperkalemia, normal potassium in patients with hypokalemia across the use of a potassium-sparing diuretic by itself, potassium-sparing diuretic with an ACE inhibitor, potassium-sparing diuretic, ACE inhibitor, and K supplement. And then the final group would be the potassium-sparing diuretic, ACE inhibitor, K supplement, and patients who are predisposed with one of the other risk factors, the diabetes, renal impairment, et cetera. And so just to get an idea, because these things tend to be additive and probably maybe more than additive because they're working by different mechanisms, we see that the risk or the incidence of symptomatic hyperkalemia increases, the absolute potassium values will increase, and the numbers of patients who have normal or hypokalemia declines over as we get more and more of these drugs that produce themselves hyperkalemia and therefore combined increase the risk. Let's go on to the next slide. Now, this whole interaction between hyperkalemia with ACE, ARBs, and potassium-sparing diuretics has been in the literature for a long time, but it actually was really brought to the fore sort of through some unintended consequences of a rather landmark study. And this is the study, a lot of you probably remember this. This is the RAIL study, and this was a study which was designed to study the effect of adding spironolactone to patients who were receiving an ACE inhibitor and a loop diuretic who were being treated for heart failure, severe heart failure. And what they found in this study, when the safety group was monitoring it, was that patients who were on spironolactone versus those not, who were getting just the ACE and the loop, were actually living longer. And so they stopped the study early. And that got a lot of attention, as you might well expect. If you go to the next slide, the thing that we need to keep in mind was that this data was collected in a group of patients under study. So these patients had severe heart failure. They had relatively few drug or disease reasons to develop hyperkalemia, so they really had eliminated those patients. They were being closely monitored so that if their potassium started to rise, they'd pick that up and could adjust for it. So this is a classic example where under the auspices of a well-designed, well-controlled study, they got a good result. Well, this got in the New England Journal of Medicine and of course was then rapidly transmitted through the prescribers, and spironolactone became a darling drug to be used in patients who had heart failure, who were receiving ACE inhibitors and perhaps potassium-sparing diuretics. This was in 1999. So let's jump forward a few years and go one slide forward to 2003. David Yerlink did a study looking at the reasons why patients were hospitalized for drug interactions. And they looked at a large population, and they actually looked at 4 different studies or interactions. Three interactions. The ACE inhibitors, people who got hyperkalemia with or without K-sparing diuretics. Glyburide, they were looking for evidence of hypoglycemia with or without cotrimoxazole, and then DIG toxicity with or without clarithromycin. And obviously the one we're interested in is the ACE inhibitor data. So if we go to the next slide, we can take a look at what they found in this trial. This was a case control trial that was done in patients in Ontario. There were patients who were receiving ACE inhibitors, and you can see there's a really large population here. And what Yerlink was interested in was patients who had been admitted to the hospital with the diagnosis of hyperkalemia. And they found a fairly small percentage of those, which is pretty typical. It's not a very common event, but it can be bad when it happens, obviously. The length of hospitalization is listed here, and 21 of these patients expired. Now, when they looked at the patients who were receiving K-sparing diuretics, either 1, 2, or 3 weeks after exposure to the diuretic, these are the odds ratios for being hospitalized for hyperkalemia compared to those patients who were not getting K-sparing diuretics. And you can see these are pretty big numbers. And for a lot of us, we're used to seeing odds ratios of 2 or 3 or 4 and getting pretty excited. These are really big numbers. So that, there's clearly a marker here for something going on. So if we go on to the next slide, Yerlink thought, gee, there must be something going on here. What's happening? And so the first thing they did was they looked at the use of spironolactone in Ontario, which is where Dr. Yerlink practices. And they looked back to 1994 and then looked forward to 2001, which was this was published in 2004. So it's just after that period. But you can see the numbers go from roughly 34, 35 back in 94. And this is the rates per 1,000 patients. That's the number of prescriptions per 1,000 patients. So started at about 34 and ended up in 2001 at 149. The RAIL study was published in 1999. And you can see the nice jump in prescription of spironolactone after that study was published, which shows that, publishing good data actually does have an effect in the way patients receive drugs that are being prescribed. Let's move to the next slide, also from Eurlink's study. Now he's looking at the admission rate per 1,000 patients for hyperkalemia. And again, the same population over the same time period. Again, the rail study at 1999, and the admission rate went from approximately 3 per 1,000 up to about 11 per 1,000 in 2001. And again, the nice increase as the spironolactone use increased over the period of time right after the rail study. So why did this happen? Well, I think that the problem here was that the people who heard about the rail study and thought, well, gee, spironolactone really does work in these patients, so therefore we should be using it. They did that. The problem is they did not monitor probably those patients closely enough, or were not as selective in choosing patients to put on spironolactone as were the physicians, the investigators for the rail study. And so there was, I suspect, a broader population exposed to the combination, which produced an increased risk for hyperkalemia. Let's move on to the next slide. A follow-up study, which came out of a different group, this is out of Germany actually. This is again a very large case control trial looking at patients who were admitted with hyperkalemia and compared the combination again of spironolactone and ACE-ARBs versus ACE-ARB without the spironolactone. And here again, they found an odds ratio increase risk of about 14. So once again, another study which really shows what they found. We mentioned earlier that trimethoprim in the Bactrim combination, or even by itself, is associated with hyperkalemia. And Eurlink also studied that, and they found that there was about a 7-fold increase in the risk of hospitalization for hyperkalemia when trimethoprim was combined with ACE inhibitors. So it also is a combination that we need to be careful with. Let's go to the next slide. Now, pharmacists probably are pretty used to seeing this, but if you prescribe potassium-sparing diuretic and an ACE or an ARB to a patient, most of the computerized clinical decision support systems that trigger interaction alerts will give you an alert. This is some data out of one institution, and this is literally the most common alerts that were triggered that require physician intervention. So I don't know what, everybody has a little different definition of that in their systems, but that's what this is. And you can see right at the top of this list is potassium-sparing diuretics and ACE and ARBs. So this is really a common interaction alert. And if you think about, that some systems are triggering thousands of alerts per month, 8% of those that's a lot of alerts. Let's move on to the next slide. So we're faced with this problem of potential hyperkalemia associated with these drugs. How do we manage this? And I think this is probably the most important slide on the whole talk, even though it's very simple. Obviously, the most important thing to do here is to monitor serum potassiums, because if you put patients on ACE inhibitor ARB plus potassium-sparing diuretic, the chances are there's going to be an increase in their serum potassium. Now, if the patient starts out hypokalemic, that might be a good thing. But if they start out normal kalemic or anywhere near the upper end of the normal range, that could end up being a patient who develops hyperkalemia. And as you probably all remember, hyperkalemia can produce a number of symptoms. And so the things to look for, of course, are diarrhea, abdominal pain, muscle weakness, paresthesia, paralysis occurs with hyperkalemia. And the other side is the cardiac effects. And I think we've all remember the peak T waves that are associated with hyperkalemia. But more importantly, of course, is you can get QRS widening, loss of atrial activity, and worst case situation, VFib and asystole. So monitoring both the serum potassium as well as patient signs and symptoms and electrocardiogram is probably the best way to get a handle on these patients. Now, the good news is that when you start somebody on this combination, the potassium change will occur fairly quickly. This doesn't take very long. The body responds quite rapidly to this, and the kidneys start hanging on to potassium, and you'll see potassium elevations in 5 to 10 days, if it's going to occur. So that's an easy thing to keep in mind. Somewhat, it's a little harder to do in outpatients than inpatients, but it's certainly worth considering. Identifying patients with disease-related risk factors or concomitant drug risk factors is also really important. And those are the patients, and we've talked about those already, who really need to have the monitoring. And even in the case of an outpatient, it's probably good to have them come back and get a potassium level drawn just to make sure that they're not slowly accumulating potassium and up until they get into a situation where they're going to have an adverse effect, which is what we're really trying to avoid. You can consider alternative therapy, although there's not a lot of alternatives here. You could try adjusting dosage downward of potassium-sparing diuretic, and that may be effective, although we really don't have data for doing that. We don't know whether we maintain efficacy or not, and the problem is we may not, but we really don't know because the data is all with 25 to 50 milligrams of spironolactone. We could think about using alternative therapy for the ACE inhibitor, but for heart failure patients, that's not real easy. You know, we don't have a lot of good options. So let's go to the next slide. What we've tried to do in this study, and the whole purpose of Dan's grant, is to develop some algorithms that help to understand and classify the parameters which increase the risk or reduce the risk to patients of developing adverse effects from these potential interactions. And those of you who've seen our webinars before are familiar with the way we've set these up, but this simply is an algorithm which passes down through a patient receiving an ACE or ARB, who then gets potassium-sparing diuretic. And so at that point, there's a decision that needs to be made, and that decision is based on what's this patient's potassium concentration. And if it's, is it less than 5 or more than 5, or 5? So less than 5 or 5 or greater within 48 hours before the start. Now, the reason why the 48 hours is in there is because we think we really need to have a pre-level. If you don't have one, then you need to get a serum potassium. If the answer to that question is no, it's not less than 5, in other words, it's greater than 5, then you go to the little red box, which really means you should probably avoid this. This is probably not a good idea. And if you're going to do this, you probably need to make sure that the benefit outweighs the risk. And the way to mitigate the risk, of course, is to take very careful monitoring approaches to the patient. If the answer is yes, concentration is less than 5, then the next question is, what's the renal function? And if the renal function is greater than 60 mils an hour, yes, then probably very little risk to the patient. These are not the people who get into trouble with hyperkalemia and end up in the hospital. On the other hand, if the GFR is greater than, is not greater than 60 mils an hour, in other words, less than 60 mils an hour, that patient needs, again, to have careful monitoring. They're at somewhat increased risk. And if the potassium levels, of course, rise above 5, they're going to be at greater risk. So the point here is, for these patients with some renal dysfunction, we want to get on the monitoring early and make sure that they don't move to that level. So this approach is an approach where you can change the values. You can change your potassium concentration from 5 to 5.5 or 4.5 or whatever you'd like to put in there. We think 5 is reasonable based on what's in the literature. But, you know, that's a number that could easily be altered. The eGFR, 60, 70, 40, 30, I mean, you know, you could come up with some number. Clearly, as the renal function drops below 60 or 70, you start to see more accumulation of potassium. And when you get down into the 30s, you really got a problem. So we think being a little more conservative here is a reasonable place to be. There's not as much data on the effect of renal function other than people, obviously, as we saw earlier, who have renal dysfunction will have increased risk of hyperkalemia. This approach, which is a very patient-specific approach to the management of interactions, is really important. It's the point of this whole grant, of course, is to come up with these patient-selective approaches, which we think are a much, much better way to deal with these potential interactions. If we go to the next slide, I have the pleasure of introducing our second speaker. This is Lorenzo Villa, and As you can see, he's from the Department of Pharmacy Practice at Mercer University, and he's going to talk to us about some work that he's done on our grant. Lorenzo, thank you.
Thank you so much, Dr. Horn. So thank you everybody for attending this webinar. I'm going to present to you this study that we conducted within our group. It's a systematic review and meta-analysis of potassium changes due to the concomitant use of ACEs and ARBs and spironolactone. So next slide, please. So the aim of our study was to systematically evaluate the evidence concerning changes in serum potassiums in individuals taking ACEs or ARBs with spironolactone and compare with those that they're taking ACE or ARBs alone. So to do that, we conducted a systematic review in using PubMed, Embase, Scopus, and Web of Science. So all of those four databases we examined, and from there, we extracted more than 1,200 articles. And then going to the title and the abstract, we eliminate a substantial number of those articles. And luckily, because I speak Spanish too, so you can tell from my accent. So we included some articles from Latin America too. And then after doing other screen in the titles that we collected, we excluded 600 studies because they were not in English or Spanish, but also because we were missing the outcome that we were looking for, which is like an increase in potassium and serum potassium. So from those 98 articles, we finally read through the entire, all of those articles, and we excluded 78, which is a big number too. And then we included 20 randomized controlled trials that they were reporting differences between those that they're using the combination or those that they're using only ACEs or ARBs. So the outcome of interest was the serum potassium variations. So, and we use R for our purpose, that package, and then we measure the mean differences. And also we control for heterogeneity, and we also assess for the result bias using mostly the tool provided by the Cochrane library. So also we did, I conducted a sensitivity analysis to evaluate the robustness of findings. And this sensitivity analysis were performed using the leave one out approach. So every time that we run the analysis, we take one study out and we run the analysis again to see if there is some variations changing or leaving one study out. So then next slide, please. So here is we have the visualization of our meta-analysis. This is the forest plot that we create. As you can see, we collect information and studies from 1995 to 2015. And those 20 studies, finally, we collect information of 570,000 patients that they use the combination, spironolactone plus ACE or ARBs. And then we have information for 547 patients that they use not the combination. So as I said before, we conducted this using the random effect model. So 2 studies that we included were conducted in North America, 9 studies were conducted in Europe, 8 studies in Asia, and 4 studies were conducted in Oceania. So, and also have to say that in 16 studies, they use the combination of ACEs and ARBs with spironolactone using 25 milligrams of spironolactone. 2 studies, they use 20 milligrams of spironolactone, and 2 other studies, they use 50 milligrams of spironolactone. So when considering all of these studies together, you can see at the very end, there is like a dark blue diamond. So the serum potassium increase with a mean difference of 0.19 milliequivalents per liter. So with a confidence interval of 0.12 to 0.26. And the results, as you can see too, they have like an intermediate heterogenetic. And because I said before, we did also seeing this variation. So also we conducted an analysis to produce a prediction interval. This was due to the large variation in the strength of the effect. So this prediction interval was calculated to help with the clinical interpretation of the heterogeneity by estimating a true treatment effect that can be expected in future study. So that was the meaning of that red line that you can see at the bottom. So considering all the 20 studies, the prediction interval was ranging between 0.06 to 0.44 milliequivalents per liter of serum potassium increase. So the next slide, please. As I said at the very beginning, also we controlled for risk of bias and the study quality that we included. So as you can see on your right, they have this like a tool provided by the Cochrane Library that you can use when you're running systematic reviews and meta-analysis of randomized controlled trials. As you can see there, in general, so the reporting risk of bias was low for 16 studies. And then you can see the overall, all of those green circles are those studies that they have like a low risk of bias. We have also 3 studies that it wasn't clear to assess all the domains. So all of those studies are in yellow color. So they have like an unclear risk of bias. And then we have one study, the red one, from 2009, that it has like a high risk of bias. And that's why we justify the sensitivity analysis that we did. So when we take out, for example, Guinea 2019 out of the analysis, so we didn't have like a significant variances in our main results. So the main results, they were really robust in that situation. So also you can see in your left, the funnel plot showing some heterogeneity of our studies included. So you're expecting here to have like a kind of mirror situation within that pyramid. Okay, next slide. And then, as I said before, so the principal findings were that the addition of spironolactone increased the serum potassium levels by a mean of 0.19 milliequivalents per liter, compared with those patients that they received ACE or ARBs alone. So even though this is a modest amount and patients taking this combination, it's necessary to monitor for potential hyperkalemia. So the key message of this study is that changes in serum potassium levels need to be assessed all the time, right? Dr. Horn say a lot of that thing, I mean, refer to the same issue at the end of his presentation. So it's always effective to use, or it's recommended to use the lowest effective doses of spironolactone in ACEs and ARB if you need to combine them. And then also, finally, the serum potassium and renal function has to be also monitored. And in our meta-analysis, when we were reading through all the articles, we found that in total, from those 20 studies that we included, we found that in total 23 patients were withdrawn from those trials because they suffer of hyperkalemia, and they were in those studies. Hyperkalemia was defined as serum potassium above of 5.4 milliequivalents per liter. So next slide, please. So now I'm going to introduce to you Dr. Richard Boyce. He is associate professor at the University of Pittsburgh. Richard, it's all yours.
Oh, thank you very much, Lorenzo. So my job on the team is to take the clinical algorithms that are developed for contextualizing drug-drug interaction alerts and turn those into logical descriptions that can be ran over electronic health records data to test how the rules will run in a particular EHR environment. Can you move on to the next slide, please? This slide gives an overview of that process. So you've seen the flow diagram, the logic flow diagram, which breaks down how the ARB and case-sparing diuretic algorithm works. What we did is then we wrote the logic for that in a logical language called rules. And then we tested it against electronic health records data. For this particular one, we used synthetic electronic health records data. So we generated patients in the electronic health records system that would match the different criteria specified by the algorithm. And the purpose of this is because we want to make sure that we can capture the different branching logic parts of the rule, given the ways that drugs and labs are coded, lab measurements are coded in the EHR data. And then what we have at the end of that is we have an artifact that can be tested by other folks, but it's been at least verified over some EHR data to have functioned. So this diagram shows that process. For our use case, we took loaded the synthetic EHR data into the OHDSI common data model. Some people might be more familiar with it as the OMOP common data model. And then again, the language was rules, and we just ran that over the electronic health records data so we could confirm that every branch of the rule would function. Can you move on to the next slide, please? So this repeats a little bit of that information and also breaks down the patients that were loaded into the common data model. Again, these are generated patient records, totally synthesized. So this was patients that would have potassium greater than or concentration greater than or less than the 5 millimole liter or molar equivalents that you just heard described. And then also, depending on the criteria, if they had an EGFR greater than or less than 60 milliliters per hour. Go ahead, next slide, please. So this is an open source project. The logic for the rule is public. It's up on a website called GitHub, and we provide the link on this slide. It'll also be posted up on, or is already posted up on our ddicds.org site. And then we include for the project something called the Docker container. And so if other groups or organizations would like to test the rule in their environment, they're able to download the Docker container and run it, and it has the entire framework for running the synthetic data and the rules in their environment. They can also change some of the configuration and point it towards data that they might have in the OMOP Odyssey common data model if they like to test it over their own data. Can you move on to the next slide, please? One of the things that we are also providing publicly is the value sets that we used in testing the rules. For folks who aren't so familiar with that, value sets are collection of codes from one or more terminologies. So in the data set that you might pull from an electronic health record, the drug exposures might be coded as NDCs, or they might be recorded as RX norms, depending on the system that's being used. And to capture, for example, spironolactone exposure, there's a number of products that are out there. So we would want to assemble a set of the codes that would be used then to identify someone who's been exposed to that. And the same thing could be applied to other clinical domains like measurements, for example, and conditions. So these value sets we created for the rules and used them in testing the rules, and then we made them publicly available. Can you move on to the next slide, please? Where you can access the value sets is on the National Library of Medicine's Value Set Authority Center. This screen, I always want to show this to people because if you just go up to the Value Set Authority Center, you'll find you need a login. This is something that's free for folks to get. They just have to go through the UMLS registration process. The reason that that's required is because some of the terminologies that are provided within the Value Set Authority Center have restrictions. For example, like SNOMED is free in the United States, but not externally. And there's other kind of restrictions for other terminologies. In this case, we're only using RX norm and LOIC, so there's really no major restrictions. Can you go on to the next slide, please? This shows what you would find if you logged in there. This is just a one, which is the value set for the ACE inhibitor or ARBs. And this is a partial listing. If you look down at the bottom, you can see there's actually 164 records in this value set. This is what's called an extensional value set. So it's a full listing of the codes. We do not have an intentional value set for this, which would be a logical definition of how to arrive at these codes. So that means that some of these codes may go away over time as the terminologies advance. So that's just something to be aware of, but it does show the thinking that went into developing these codes and the set of codes that we agreed upon. Go ahead, next slide, please. So as I mentioned before, we have a public website, ddicds.org. And if you go up there, actually just land on the front page and go to the drug interactions tab, you'll be able to find the page that's specific to this algorithm, or you can use the link in the slides, which will be available to folks. And down towards the bottom of the page, after a very detailed narrative that was written by one of the folks on our team in collaboration with the other clinical experts, you'll find links to these things, including a PDF of the logic flow diagram, the Joules code, and value sets in various formats. And so we hope and encourage folks to go and check those things out and then contact us with any questions. Next slide. Okay, I'm done. Passing it back to you, Dan.
Well, thanks, John, Lorenzo, and Rich. So this is the formal presentation that we had prepared this morning for you. Across the attendees today, I imagine we have a variety of different stakeholders, and those include individuals who help curate the knowledge associated with drug interactions, but also individuals who probably are in front lines in terms of implementing rules associated with drug interactions. So I'd like to get a sense from the audience of to what extent have you implemented a rule associated with this particular drug pair? I know some of the EHR systems allow some of this to occur. So if you've done that, and you'd like to discuss how it's going with respect to how you implemented it and in what environment you've implemented it, and then any responses to the implementation, sort of tailoring this, probably based upon potassium levels. We'd like to hear that conversation. The other thing I think that's unique about today's webinar is the notion that we've been trying to figure out at what point do we have cut points for potassium or kidney function. You know, most patients who receive this combination probably do not develop hyperkalemia. We don't know that for certain, but we gather that from the evidence that suggests that there's a relatively modest increase in potassium values. So what we do need to worry about are those patients susceptible to this interaction, and Dr. Horn highlighted the various other risk factors. But obviously the monitoring potassium is probably the most important way to prevent patients from from developing hyperkalemia. A little bit more challenging in the ambulatory environment as compared to, I should say, the inpatient environment. But is there anybody who would we're happy to open it up for discussion if you'd like to make a comment? Feel free to let us know via chat or raise a hand, or feel free to open up your microphone. I think we've opened it up to all participants now, haven't we, Sheila? Or I guess I will, yeah, if you let me know, I can allow you to talk.
Looks like Radha Manian had their hand raised. I've unmuted you. Ask your question or make your comment.
I'd like to know what are the actual logistical steps involved in implementing these rules and putting this algorithm, actual logistics of putting the algorithm into the EHR, or can it be attached to the database in a pharmacy database?
Rich, I think that's a question more directed at you. So how could the rules code be implemented if one were interested in implementing that? I think.
Okay. Yeah. So the question's great. The rules code that we were talking about is designed to operate off of a specific representation of electronic health records data. So that's the common data model that I referred to. And what I mean by that, has a specific set of tables, and those tables have a specific set of columns, and then they use a particular vocabulary. So the rules are written to operate on data in that format. And the OMOP common data model is not like a live EHR database like Epic or Cerner, which is a highly transactional database. Those systems and other ones like it, they're going to have some kind of a rule implementation process. Sometimes they have even a web-based, form that people can use to design the rules. And so the purpose then of what we did is it gives a very concrete specification for how we did it. And then as people try to translate the flow diagram into the tool that they use to write rules in their electronic health record system, they're going to come up with questions. They're going to be like, well, what do you mean by spironolactone? Or what do you mean by, the timing, for example, how far do you look back, to determine the lab? And what do you mean by concomitant exposure? So then they can look at the logic that we wrote, and it gives a concrete specification for that. And they can look at the value sets, and it gives a concrete specification for that as well. So really the fact that we built that and that it executed is for the purposes of having a real, like a reference implementation of the rule. And most likely in an actual real world scenario, someone's going to have to go through with their team using whatever tools are given by the EHR system, the whole process of re-implementing those. We are working on shareable artifacts for this. So these artifacts are written in a language called CQL, clinical quality language. And that's something that the Agency for Healthcare Research Quality and Funding and Medicare, CMS, and some other organizations within the HL7 community are really promoting. And those artifacts will, in principle, be implementable by anybody who has an engine that can run those within their site. Or another approach would be for us to set up a service, for us or another organization to set up a service that would have the reasoning component, and then using something like CDS hooks, pick a point in the workflow where the rule should trigger. So like maybe when the physician is selecting a medication or right when they're signing it, and then using the CDS hook standard, it can call the service, pass on some information in a secure way to the service, and get a response back with suggestions like change the medication or reduce the medication. Those artifacts are not developed yet for this specific interaction, but if you look at some of the other interactions, we have developed them, and we have an implementation guide for how that CDS service should operate as well. So I hope that helps to answer your question without going into too much detail to overwhelm you. But does that help a little bit with the answer to that question?
Yeah, I got a kind of overall view of it. Maybe I will get in touch with you later to get some details of the steps. Thank you.
Thank you.
Looks like I have another comment or question from Christine. Helena? Let's see. Maybe people are inadvertently, Dave, go ahead. David? No, I think you should be able to speak if you just unmute your mic.
Yeah, I'll just say our local ministry that I work for not too long ago put in several alerts during order entry for elevated, electrolyte levels, drug levels, when a provider is placing an order for one of those products. We've also, and that includes potassium. So if a provider is ordering a potassium product for a patient with a potassium of 5 or higher, they get a notification, and that includes potassium-sparing diuretics. Nationally, we've recently approved, shutting down the standard med vendor ACE inhibitor, potassium-sparing diuretic drug-drug interactions, in combination with the alerts we have in place for patients with the potassium over 5. And additionally, we're going to use a third-party clinical monitoring system to identify patients with potassiums over 5. So kind of doing some of the same work to try to minimize that. Locally was our number one drug-drug interaction that we were seeing. Had well over a 98% override rate. Just really didn't mean anything to the providers that were being bombarded with that interaction alert.
Yeah, that's great. You're using, you said, a third-party monitoring program, so that's pulling information out of the EHR on a daily basis and providing a dashboard for the pharmacist to intervene on if necessary.
Yes, sir.
Okay, all right. And did you have any pushback with establishing the five as your cut point, or how did you go about deciding that was the level that you want to trigger your third-party program to send a warning?
Yeah, that was just a decision made, locally with our medical staff, and just kind of looking at our local, normal ranges for potassium. 5 is the upper end of that normal range for us, and that was the decision that was made.
Okay, well, it's obviously supported by the data, and that was one of the pieces that we wanted to make sure that, when we were putting our algorithm together, that we try to be as evidence-based as possible. So that's why Lorenzo and others have undertaken this systematic review to kind of come up with, I guess, some level of comfort about using 5 as a cut point. So if we saw significant increases in potassium changes with the addition of the combination, I think we would have been a little hesitant to use a value of 5. We would have maybe argued for something less, like 4.5. But 5 seems to be reasonable, given that most patients probably are not going to get in trouble if they're close to 5 and you add the combination. They're not going to get over 6, and certainly probably not going to get to 7. So as long as they don't have other risk factors. So that's the challenge, is to bring those in. And to be fair, we decided not to include those other risk factors in our algorithm just because we feel like potassium monitoring is probably the best approach, along with obviously kidney function. But those two pieces together are probably sufficient versus trying to drive into all of the other potential risk factors that John had indicated. So, other comments or methods that people have done with respect to this particular interaction that they'd like to describe? And you should be able to unmute yourself and feel free to speak. Okay, well, I'm not hearing any, and I don't see anything in the chat at the moment. So thank you all for attending today's program. We won't go on past the hour. We appreciate your attendance, and we hope that you'll be able to join us for one of our future webinars. At this point in time, we don't have one scheduled, but we'll notify you when we do. So best wishes for your success in your institutions or organizations. And thank you again for attending. Have a great day.